for the opportunity to present tonight at the gala. And I'm Lois Cohen, formerly Associate Director of the NIDCR, National Institutes of Dental and Craniofacial Research at the NIH for Global Health Research, a number of other positions I held before, and currently am the Paul G. Rogers Ambassador for Global Health Research and a consultant to NIDR and on the Shils board. And it's my honor tonight to present the 2020 Innovation Award for Research to a colleague in our own institute in our intramural program at the NIDCR in Bethesda, whose research has been very central to the COVID-19 pandemic and its mitigation. Blake M. Warner has a Bachelor in Biochemistry, an MPH, a PhD, and a DDS, and is a dentist scientist, oral pathologist in our intramural program, who has been leading since August 2019 the salivary disorders unit in our Sjogren's syndrome clinic. He leads a multidisciplinary clinic focused on disorders of the salivary gland. So this research that he has proposed and has executed turns out to be very important in today's world of COVID-19. And he had proposed a unique project to the NIH. It was reviewed and accepted on the transmissibility and viral load of the coronavirus in oral secretions. The objective was to determine if the virus is present in oral secretions of COVID-19 positive patients. And he designed a study to determine if using masks can prevent the transmission. And this study has greatly informed our community on how COVID-19 is spread among people and how it can be presented. So it's with great pleasure that I have the honor to present Dr. Warner with the 2020 Innovation Award for Research, and let Blake himself give you some insights into his findings.
Sure, thank you so much, Lois, for that introduction. That was wonderful, and I'm very humbled to receive this honor. So thank you to all those who were involved in the nomination process as well as awarding this award. Yes, I'd very much like to talk a little bit about how we came to this. And I think that being in the intramural program afforded some possibilities that might not have been available to me if I was working in the extramural environment, including the interaction with colleagues that are specialists in virology, that are specialists in executing clinical trials and clinical research studies, and then having on hand a phenomenal clinical research team to help me construct not only the clinical study, but also these very the specific aims to help understand the dynamics of SARS-CoV-2, the virus that causes COVID-19 disease, and how it can be transmitted through oral secretions. I want to go back to how this all started. This started with a conversation in mid-January with my research mentor, Dr. Jay Carini, who's a virologist. And we had this thought, this conversation, and this is how things happen in science. Sometimes it happens in the hallway. And we said, you know, this is going to be a lot like SARS-CoV or SARS-1. And there's a possibility that this virus uses the angiotensin converting enzyme 2 as a target to enter cells, or an entry factor. And Jay kind of asked me, do you know if it's expressed in the salivary glands? And I didn't know the answer right off the top of my head, but I did have results from a technology called single cell RNA sequencing, where we take a tissue such as the salivary glands that we procure in the clinic, part of my Sjogren's syndrome clinic, and you break them up into individual cells and you sequence each one to look at all the genes that are turned on or turned off in each individual cell. And then using that cell's transcriptome, you can map, basically show which cells express certain entry factors. And we did that off the cuff sort of calculation. And it pointed to the salivary glands, in fact, likely to be vulnerable to SARS-CoV-1. And right then we said, you know, it would be interesting if we could get a hold of some saliva to see if in fact saliva was infectious. For me, it's what I've been doing for the last 7 months, every day, 12 hours a day.
How exciting and how important to the nation and to the globe for that matter. Just a question, what kind of mask? We have so many masks out there. I assume,
Sure.
Were you using N95s or?
We were not using N95s. You know, N95s right now are in short supply, and they're really only useful for healthcare providers, right? Where you're going to be exposed to potentially infectious aerosols. They're intended to reduce aerosol transmission. But just a simple cloth mask, and this could be the one that you receive, let's say, when you go to the doctor's office, or it could be one that I use when I'm driving to work, which is a cloth mask that really just covers your mouth and nose. Right. Both of those tend to reduce the amount of droplets and the velocity of those droplets when they shoot out of your mouth, because even if we don't think we spit very much when we talk, we all do, some more than others. And it really is about interrupting that path, okay? So you have a cotton layer or some sort of fiber, it seems to both disrupt the ejection velocity, but also can capture some of those larger droplets. And so I would say that any mask really will do. The only masks I advocate against are ones that are ported or that have little valves on the side.
Yeah, I can breathe better through those.
Yeah, but they help. Yeah, unfortunately, they help no one. They don't help the people who are using to be more protected, and they don't help others to be less likely to be infected. So they help you breathe better, but I would say that the effort that you have to breathe through a cloth mask is it's very tiny difference. So.
Well, thank you. That's very, very exciting. And we're very proud of your work. Not only the Shils board, of course, but of course our whole NIH is. So thank you, Blake.
Yeah, so thank you to the Shils board for this award. Thank you to Larry Tabak for nominating me for the award. And thank you to basically my whole team for helping get this done. It really is an award that I'm receiving on behalf of my team, because I couldn't have got any of this done without them. They really do keep this machine well-oiled and moving forward. And I'd also like to thank my collaborator at UNC. His name's Kevin Bird. And maybe for the last 4 months, he and I both were interested in this question. And we've been working in tandem most hours of the night to try to get this data disseminated. And I think it exemplifies the best of NIH, the best of dental research. He himself is a DDS PhD that kind of came up in the same cohort as me. And it shows that really dentistry can make a huge difference, not only for US public health, but for world public health as well. So thank you.
Well said, well said.